Right. So, hello, and thank you very much for this opportunity. I come from single molecule detection, and what I'm trying to do is apply these techniques, which are basically coming from the realm of physics, to biological systems. And the underlying principle is the fact that when we want to measure a property of something, usually a population, when we measure it on the whole population, we get an average property of the whole population. So we cannot distinguish little differences and variability within this population. And this is true for most of the physical measurements that we do in science. And I'm trying to do this on genomes. So this is the human genome, a very complex system, about 2 meters of DNA in every cell in our body, and many, many, many miles of DNA from here to the sun and back in each one of us. And there's a great degree of variability in our genomes. This is why we're different. And these differences, many of you have heard of them. These are either what are called single point mutations, or SNPs, single nucleotides, single bases and letters that are different between us, or structural and copy number variations, which are larger chunks of DNA that differ between individuals. There could be multiple copies of the same region in the genome, and the number of copies is different between people. Or it could be segments of the genome which are inverted, missing completely, duplicated, mislocated, all kinds of things. Despite of that fact, our genetics as humans, as a species, are very, very, very similar. We actually share 99.5 to 99.9 of our genetic sequence. And yet, we are different. Not only that, if you take two cells from the same person, these two cells share exactly the same genome. And still, they are very, very different. And this is because there's another component called epigenetics, from Latin above genetics. And this is a whole layer of information that rides the genome but is not encoded in the actual 4 bases of DNA. It's like an annotation to the genome, and it's a very, very exciting area, which is relatively new. And within this topic of epigenetics, which is complex, I concentrate on chemical DNA modifications. So these are actual chemical reactions that are performed on our DNA bases by molecular motors in our cells, which usually add a little tiny molecular moiety to one of the bases, and this way completely changes the properties of these. Now, these processes are very dynamic, and therefore we're expecting them to be very variable between cells. If they're variable and we want to track them down, it's very important that we look at genomes on the single molecule level. Okay? We want to look at them one by one. Now, the ways that we have at our disposal to look at individual genomes today are either by cytogenetics. These are the traditional genetic tests that many of us have had just before we were born. And we look at intact chromosomes. We know how to paint them and extract information from them. But the problem here is resolution. This is an optical measurement. Even the tiniest feature here encompasses millions of bases. And this is because at this stage, when we're looking at it, we have 2 meters of DNA condensed into a nuclei of 10 microns. So every little feature here is huge, and we can only detect very, very large differences between genomes. On the other end of the spectrum, we have sequencing. So sequencing has the ultimate resolution, a single base pair. It can tell us the composition of DNA base by base. But many people are not aware that we can today only take a sequence of a very short piece of DNA on the single molecule level, about 400 bases, 400 letters. Now, it turns out that a very, very interesting genomic domain is between 1,000 and 1,000,000 bases, because this is the typical length of our genes, of the pieces of our DNA, which are the most functional pieces of DNA encoding for proteins. And what I'm trying to do in my lab is basically position myself here and try to bridge the gap between these two realms of genomics. And the way that we do this is basically by unraveling individual DNA molecules and looking at them on a linear axis. What we see here is that we use these nanochannel arrays, arrays of very, very thin channels, where we can squeeze DNA through these channels and this way align the DNA molecules to a linear form. What we see in this little video is a 400,000 base pair piece of DNA. It's passing through an array of pillars that transforms it from the micro regime to the nano regime, and then the DNA enters the channel and straightens up. This way, we have the potential of reading information along this piece of DNA in a contextual manner. However, at this stage what we get is a lot of green lines. And they're green because we know how to paint the DNA fluorescently in a non-specific manner so that it appears as a fluorescent line. But there's no information here. If we want to read information out, it would be very nice if we could generate some kind of barcode on these molecules that can identify where they come from in the genome. And we know how to do this. We know how to use enzymes in combination with chemistry in order to highlight specific words in the genome. And the arrangement, the specific arrangement of these words is unique to every region in the genome, just like a book has a specific arrangement of words in every page. So when we do that, it's a little bit hard to see with this contrast, but we have many, many blue lines in this case, which are the DNA, and these lines are dotted with green spots, which are this barcode that tells us where these molecules come from in the genome. Now, if we want to look at differences, we obviously need to measure many, many molecules that represent the same location in the genome so that we can compare them side by side and look for differences. And this is basically what we're seeing here. This is an example of a measurement performed in the lab where we see many, many of these DNA molecules passing through these nanochannels as we take pictures of them. And we can generate a lot of statistics this way. And then in order to use this information, we can use image processing to extract the coordinates of the dots from the DNA molecules and then check their location on the theoretical file of the genomic sequence. So we can take each one of these molecules and kind of slide it along the genomic pattern and look at where they fit, just by pattern recognition. And this is an example where a piece of DNA clusters in a specific region, and now we have the opportunity to compare between individual molecules that arise from the same genomic location. Now what we can do with this, and what we see here, basically every yellow line here is a molecule that was detected in the microscope, and every blue dot is a fluorescent spot on this molecule. And first of all, we can see now DNA repeats, these structural variations that we were talking about. So you can see here that many molecules have this array of repeated spots, which mean that we have the same sequence of DNA repeating itself many, many times. And many diseases, for example, arise when we do not have the right amount, more or less, of this repetitive element. Another very interesting thing that you can do with this, just looking at the sequence, is what's called haplotyping. So each one of us has a copy of his father's DNA and a copy of his mother's DNA. This is why we have 2 copies of each chromosome. And they're different. Each one of these gives us our mother's faults and our father's faults to be combined. And for example, here we find 2 groups of molecules which are almost completely identical, but this group is missing a segment that exists in this group. And these are the 2 haplotypes from the mother and from the father. This is something that is very, very difficult to do today using conventional sequencing techniques. Now what I'm most excited about, as I told you before, is epigenetics. And we have the opportunity here to use color. Since this is a fluorescent experiment, we can use color to label different kinds of information with different colors. And this way, basically do the same experiment, but look at very different observables on the same molecule and correlate between them. Now, the first target that we had was called hydroxymethyl cytosine. It was discovered about 2009, so it's very, very fresh, that there is this specific modification of the C base. And we don't know much about its function, but we do know that as opposed to DNA methylation, which is the most famous DNA modification, this one appears at pretty constant levels across different tissues. Hydroxymethyl cytosine appears at very high levels in the central nervous system and in the brain, and then at much lower levels, but tissue-specific levels, in different tissues. What we try to do is actually develop a chemistry that can highlight these hydroxymethyl bases. This is a chemoenzymatic technique. We use an enzyme that can specifically recognize these modified bases on DNA and attach a sugar molecule to it. And then if we feed it with a modified sugar that has this N3, which is an azide, it's a functional handle that we can attach specifically to these bases, we can then come and attach a fluorophore to that. And as an example, for example, this is a genome of a virus where we've planted specifically these hydroxymethylated bases, and using our chemistry, we can light them up and get an optical contrast for their locations. Now we have an opportunity to basically simply count the number of dots on a piece of DNA and measure its length, and this way get a very quantitative measure of how much hydroxymethylation we have in a given sample. And here's an example, for example, from DNA extracted from mouse brain versus mouse kidney, and you can visually directly see that there's much more hydroxymethylation in the brain versus the kidney, and this is the bar graph that represents this. We've recently extended this technique to a multi-well plate format, and now we can do hundreds of experiments simultaneously. We use this to really profile the levels of hydroxymethylation in different mouse tissues with very good results and very, very high sensitivity. But of course, the ultimate goal here is to do some mapping, because we have the opportunity to actually look at epigenetic patterns on the genome. Here we have the same engineered virus DNA, and you can see that we do have a reproducible pattern when we flow these in the nanochannels and align them. So we can detect patterns, and very, very recently in our group, wow, very hard to see what's going on here, but believe me when I tell you that these are many, many DNA molecules, and they have two sets of dots on them, red dots and green dots. The green dots tell us where in the genome these molecules come from, and the red dots tell us what the distribution of hydroxymethyl cytosine in this region is. And right there at the top is a piece of DNA coming from my blood, a very long piece, about 400,000 bases, which is, I think, the first ever human epigenetic mapping. And I'm really excited about this result. I'm not always late. I was late once. Come on. It's very easy to ruin your reputation. Very easy. Okay. Now, what's up with time? Should I? Okay. So I think I'll wrap it up here. I did want to tell you about one more thing, but I know we're late and we all want coffee. So I'm going to stop here and thank a really, really great team that I have in my lab and great collaborators that allow me to do this science. Thank you very much.
Questions? What of the sequence? How to print?
What?
How to print? So after use, the surface becomes.
So,
Cannot use it again.
in principle, it should be possible to flush these channels out and clean them. It turns out, because they're very, very narrow, on the order of 50 nanometers, they clog easily and they contaminate easily. And the second experiment is never as good as the first experiment. So there's a big effort that we want to do in fabricating these kind of devices so that we can do more elaborate.
So you want to fabricate, you want to repeat the use. That's less necessary.
Yes. That would be great.
Are you flushing this into a single DNA fragment or?
No, the DNA is fragmented.
Yeah, fragment, but I mean you can use the recipient enzyme to cut them off.
So DNA,
Are they? I mean, each line is it?
each line is a single molecule of DNA. Yes, up to about 2 megabases of DNA per molecule.
If I give you a 400 kb fragment DNA, you can tell me where the methylation is and where
Exactly.
the SNPs are.
Hopefully.
Right.
Can't guarantee. It's work in progress.
So what's the advantage of this technology versus existing next gen or whatever?
So let's say you have cancer developing, and you're at the 1,000 cell stage of cancer. Not you, of course. That's the hypothetical human. So using conventional sequencing, if there's an epigenetic signature of cancer, you will be able to detect it only once there is significant population that will show up above the average. This technique, since I'm looking at the molecules one by one, it could be 1 in 10,000, it could be 1 in a million. If it passed through the microscope and I detected it, I know that there's something to look after. Okay, so basically the main advantage here is that I'm looking at populations, but on a single individual basis, and therefore I can track down very small subpopulations.
Thousands of cells?
I'm taking, yes.
Oh, rather than a single DNA from a single cell.
I am taking thousands of cells, but every one of these molecules comes from a single cell. So I do have single cell capability because if I look at a region of the genome, I have many, many molecules coming from many, many cells, and each one of these molecules comes from a different cell, and it represents what happens at this location in the genome.
So you see people make sensors with covered on tube to sense molecules, right? And they can also have single molecular sensitivity. The problem is that the chance of that single molecule attached to the sensor is really, really low. You have to wait like thousands of years or billions of years for that to happen. You only have one molecule in the solution. So I mean, I'm wondering, suppose in your body, not your hypothetical.
Yes.
I mean, there are some cells. How would you do things to define those?
So the nice thing about this technique is that it's a very, very highly parallel technique. Okay? I sample billions of molecules in an experiment. Okay? And each one of them is detected, and then the computer does the work for me for screening them and looking at different features. So it really amplifies the probability of me finding something.
Okay, I think given the time, thanks Yuval again.
Thank you.
We can discuss. Thank you.